So the research I'm doing in this lab is focused on sagebrush, which is super important in our local biome and community. We're looking at its phenotypic capabilities and its kind of genetic toolbox to see if it can withstand climate change in the future and all this incoming drought.
This research will actually benefit both local and regional, as well as possibly even worldwide populations in economic sense, that we are headed into more heat and drought. And this research is going to help us find the genes that are going to underpin drought tolerance in sagebrush plants. And then that research can also be applied across other ecosystems and other plant life.
Right now, more than 58 million people in the Western US are suffering from droughts, like all of those people. And we, by studying how can plants cope and adapt to rapid climate change, how can we make our landscape sustainable to this, therefore maintaining the water regime and all of those services that Idaho has to offer. One sagebrush, it might take years for seeds to happen. So what we have to do is say, if you remove evolution from the equation, is there a sagebrush here that can adapt to what's going on right now, that is already adapted, sorry. And then if we find this one, can we propagate or favor it so that the whole ecosystem can be resilient?
My specific role in this lab is to create the protocols that are needed for the biomass production of the sagebrush for the genome sequencing, as well as produce clones so that we can perform experiments to link the genome to the phenotypic response of sagebrush plants. So the students in the lab are doing different work than I do. They are taking epidermal peels of the outside of our sagebrush plants, which we have in our greenhouse, that are a representation of the distribution of the sagebrush steppe ecosystem. And they will do the epidermal peels and actually be measuring the stomata, as well as the opening and closing and the apertures.
I think my research work is very unique because of the way our lab is structured. We do everything from top to bottom. I'm helping make all of these plants grow. We're extracting all of the DNA, going through the experiments, and then doing all the computer work as well. So I think it's really unique that I get to be part of this whole vertically integrated process, which I don't think I've seen in other programs, or at least from what I know my other colleagues are doing.
Students are the engine of research in a university setting, and they really had a big contribution to this research. And I like the way they think and how they challenge you to be a better mentor and be a better scientist.